Alright, hi everybody. Today we're going to talk about data analysis and graphing. This video is going to be a little long, but just please make sure you understand these science practices. So science practice 4A and 4B, which requires you to construct graphs with correct orientation, labeling, units, scaling, plotting, and types and trend lines. And 4B requires you to describe data from tables or graphs and describing the relationship between these variables. All right, so let's get into data analysis. So experiments rely on the collection of data. You have both qualitative and quantitative data. We're going to talk about those in a second. But each value collected in an experiment is a piece of data. And all data points together are the sample size of your experiment. So why does sample size matter? So more data equals bigger sample size, and bigger sample size allows better statistical analysis. So better statistical analysis provides better support for your conclusions that you draw from the data. All right, and so there's different categories of data. We have qualitative data, like we mentioned, and these are observations that cannot be numerically expressed. And qualitative data is not appropriate for scientific research because it cannot be statistically analyzed. And we have quantitative data, and quantitative data consists of numbers. So discrete measurable units that are numerically expressed. And quantitative data is required for scientific research. So remember that science requires numbers that you can analyze mathematically. All right, so let's get into the data analysis requirements. So before analyzing data, we have to determine what's normal for our data sample and the population. So you can calculate the mean, mode, range, and standard deviation only after you establish normalcy. And then you can compare your data points and confirm any trends and perform statistical analysis. So always remember to calculate normalcy first, and then you can calculate, you can do your analysis. All right, so let's get into data tables and graphs. So the anatomy of the data table. So here you can see that your independent variable is in the first column, and then your dependent variable right here is going to be organized by trials, which are then averaged and calculated when experiments are repeated. Your title is going to describe what is being measured, and always remember to include your units and everything. Alright, and this video just basically tells you how to spot misleading graphs. So go ahead and watch this on your own time. Alright, so a key term you want to remember is LUTs when graphing. And so L stands for label your axis, U is use the right graph type, T is to title your graph, E is error bars. I'll talk about error bars in a second. And then always scale your data. We're also going to talk about scaling. Another acronym you want to remember is dry mix, and we'll get into that in a second. So dry mix, the D stands for the dependent variable. The R stands to the respond to the change, and the Y means that you're always going to plot your dependent variable on the Y-axis. For mix, M is what you're manipulating to cause a change. Your I is your independent variable, and remember that's always going to be on the X-axis. So remember your independent variable is what you're changing. So you want to see if this variable is going to cause an effect on the dependent variable. All right, and here's a summary of that. And again, always remember to label your units. All right, so we have different types of graphs for different types of data. So here you can see that a line graph is for continuous data, and scatter plots are to show relationships. So I always try to remember if it's time, use a line graph. And so for scatter plots, you want to explore associations between the two variables. So usually one variable is measured against another, and you'll be able to find trends and associations with this type of data. All right, so then we have bar graphs and histograms. So bar graphs are for categorical data. So this is only used when your variable is when one of your variables is qualitative and histogram is for frequency data. So how your data is distributed and displayed. So meaning you're going to basically show the mean, median, and mode. And here's more information on that. Go ahead and read it on your own time. So bar graphs versus histograms. So bar graphs show categorical data with gaps between bars. And so here, for example, you can think of things like insect types and then compare the insect types with numerical values. And histograms show frequency with no gaps between, like for example, age numbers. So remember that bar graphs are if you have different categories, and histograms means that you're gonna be graphing frequency ranges with no gaps. All right, so let's get into pie charts. Pie charts are used to represent parts of a whole, and box and whiskers graphs or plots, you can also call them box plots, display 5 number summary of any data set. And what I mean by that is you're always going to have your minimum, your lower quartile, which was represented by Q1, your median, your upper quartile was represented by Q3, and your max value. Your minimum value is the smallest value in your data set. Your lower quartile is the value that 25% of your data falls below. Your median is your middle value that splits your data in half. Your Q3, your upper quartile, is the value that shows 75% of where your data is falling. And then your max is your largest value in your data set. Again, this is what pie graphs are representing. Log scales are used to represent either really large numbers or really small numbers. So for example, survivorship curves, restrictive enzyme analysis, cells, the amount of cells that are growing throughout time. Using the right type of graph. So we're going to discuss bar graphs and line graphs. So bar graphs are used when the independent variable consists of experimental groups or categories. So you're often going to be using averages for each group. And so examples can be like different treatments, different species, different experimental conditions. So here with exercises versus without exercise. And then line graphs are going to be used when the independent variable is continuous and numerical. Again, if it's time, use a line. And this can be used to calculate the slope, which is rise over run. And you're only going to connect the points when you're showing a continuous relationship. If you want to find the overall trend, you don't want to connect the dots. Instead, you want to do a line of best fit. So you're going to find where your line can fit and capture most of your data sets. All right. Okay, so how can you title your graph? Your title should always describe what is being shown without drawing any conclusions. So you're not going to be explaining anything here. You're just going to describe what is being shown. All right. So graph titles are not worth any points, but there's 2 general formulas when you're using your graph, titling your graph. So usually you could do your independent variable versus your dependent variable if you are unsure if there's any relationship. And then you can use effect of independent variable on the dependent variable if you suspect that there is a relationship. So for example, you can say light intensity on plant growth or something like that. All right, so let's get into error bars. So what are error bars? Error bars show statistical variability and help compare significance between groups. All right. So the data points within a sample, and sample means between experimental groups. What is variability? Variability is the uncertainty of a data point. So this helps indicate the possible range within which the true value of the measurement or estimate might fit. So for example, here you can visualize how much error or uncertainty is associated with the data you collected, and it could provide more insight. All right. When the bars do not overlap, then this can indicate statistical significant differences. And we're going to get more into that in the other video. But when they do overlap, then significantly, then it means that your differences may not be statistically meaningful. So for example, you can compare phosphate to oxalate. The values don't really overlap. So that means that you can predict that there is some statistical difference between these values. But phosphate and uric acid, you see how these values overlap. So then you can predict that there is no statistical significance. All right, let's talk about proper scaling. So all your graphs must have a proper scale for the x and y axis. So you usually want to start at 0 when you're graphing unless told otherwise. And you want to make sure that you're using equal intervals between scale marks. So for example, here you're going by 10 each time. All right, and so how can you make sure you're using up your entire area of the graph? I'll give you a formula in a second. And so here is the formula. So you're going to want to use the highest number on your y-axis divided by the number of squares on the axis. Alright, so here's the formula. So here you can see your highest number is 70, and the number of squares, if you count 1, 2, 3, 4, 5, 6, 7. So 70 divided by 7 is 10. So you're going to go by 10. Should exceed your largest data points. So for example, if your largest data point is 60, then your max value here, you want to have it around 70. And this is going to allow for you to actually extrapolate information. And for example, if you have error bars and you want to go higher up. Alrighty. So think about this question. Which type of graph would you use? Like for example, this is one scenario, another scenario, and a different scenario. This is the growth throughout that's being recorded daily. Here you're seeing the absenteeism throughout time, and here you're measuring the number of cells throughout time because these phases take a while. And go ahead and think about it for a second. And you would want to use a line graph, right? Remember, if it's time, use a line. All right, now let's get into slope. So when calculating rate from a graph, you want to choose points far apart from your line of best fit. So remember, we're saying if you want to calculate, you want to do a line of best fit. And so you want to make sure that the points you choose are where your major grid lines cross. So for example, here you see a cross. And you don't have to use points from your data table. So for example, I can choose this point even though it's not specifically something I have accounted for. All right, sorry. And so the formula is your rate and change of your y over your change in your x. So for example, here you want to do y2 minus y1, right? That's calculating the change over x2 minus x1. And so here you have an enzyme practice problem. And so this slide shows an enzyme reaction rate experiment. Go ahead and pause it. But 10 milliliters starch solution plus 1 milliliters enzyme solution, and you're measuring maltose production every 10 minutes. All right, so look at the data table and the instructions to graph the table and calculate rate between 0 and 30 minutes. So go ahead and pause the video and work on it. All right, and this should have been a line graph because it's talking about time. And so the independent variable here is going to be time. So remember, it's dry mix. So all right, I got cut off, but when knowing where to put your variables on which axis, always remember the acronym dry mix. And so for mix, meaning the independent variable is always going to go on the x-axis, and your independent variable here is time. And then dry is you're going to put your dependent variable on your y-axis. So your dependent variable here is maltose, because you're seeing how maltose concentration is going to change over time. And here you're being asked to calculate the rate. And so remember, for rate, you're always going to want to calculate the slope. So your slope is always going to be your change in your dependent variable over your change in time. And for change in time, remember, change is just calculating the difference. And so the point I chose here is 10.4. So around 10.4, you have a value of 30. So 10.4 here and 30. And then another value I chose was 0. So 0 over 0. And then you're going to calculate the difference. You're going to subtract. So your y2 minus your y1 is 10.4. And then your x2 minus your x1 is 30. And then you're going to divide that. And it's going to give you 0.35 micromolars over minute. All right. And so let's get into graph analysis. So how do you analyze a graph? There's really no steps on how to analyze a graph, but I will show you how to draw conclusions based on your graph. All right. And so, for example, here we have a graph representing how cell phone use affects final grade levels. So the first thing you want to do is you want to identify the scientific question that is or could be answered by the graph. So here you can tell that you're measuring how does cell phone use during class affect your student grade. And so then you want to make an answer to your question, right? You want to make a claim. You don't want to explain anything, you just want to answer the question. And so here you can see that students that use their phone in class have lower grades than those who do not use their phones in class, right? So when they never use their grades, their grade is higher than someone who uses their phone in class, right? And then you want to look for your evidence to support your claim. And remember, here you have quantitative data, you have numbers, so you can be very specific here. So you can say the graph shows students who never use their phones have grades of 3.25, while those who use their phone over 5 times have grades of 2.8. So it's showing you that students who do not use their phones have higher grades, right? And then you want to find some reasoning. So explain how the claim and evidence are related. So why do you think that is? Why do you think that pattern is existing? And this is an example for phone, but when you do reasoning in biology, you want to talk about the science behind it. So like the biology, the chemical, the physical principles behind how your claim and your evidence are related. So one of the reasonings you can say is students using phones are not paying attention as much as those who do not. So they will not learn the material as well, leading to lower grades, right? So you're coming up with the why. And remember, for science, for biology, you're going to talk about the science behind that. All right. And so here we're going to practice. You're going to have a couple of practice problems in order for you to identify what type of graph to use. So read the question and then graph a given set of data. Then you're going to graph it. And then you're going to make a conclusion based on graphs and charts. Again, I want to remind you for the AP test, you don't want, and then you're going to make some predictions. But for the AP test, you want to remember that you're going to have, you're not going to have that much time. So try to focus on the data set. Sometimes what you read up here, you don't really need to understand it. You just want to graph what you see. And here there's a diabetes practice problem. And so here you're seeing how diabetes, here's a lot of background information, how diabetes and the glucose level is affected throughout time. All right. And so first off, what type of graph do you think you're going to be using if you're measuring how glucose levels changes over time? So think about it. And so your first step is you want to make a line graph. Remember, if it's time, use a line. And then it's asking you what evidence you have to support your answer. So here you want to be specific and use quantitative data. So you want to use numbers. And then it's asking you to make a prediction. So go ahead and do that. And now we have a practice problem with photosynthesis and water depth. So here they're measuring the amount of bubbles being released in a given minute based on the depth. All right. And so the reason they're calculating the oxygen bubbles is because in photosynthesis you are releasing oxygen. And so that's why they're calculating the bubbles, all right? They're releasing oxygen, so that's a gas, right? And so what type of graph do you think you're going to be using if you're doing bubbles released in a minute throughout water depth? Think about that. And you are going to use a line graph again for this example. Remember, if it's time, use a line. So again, this is continuous data. All right, now you have a third example of Daphnia, which is a water fleece, and their heart rate based on different stimulants. So you have different stimulants. So this is a category. What type of graph do you think you're going to be using for this? All right, and so you should have said bar graph because it is a categorical data. So remember that categorical data means that there is existing groups or categories. And the best way you're going to want to represent that is either bar charts or pie charts. And then for continuous data, you could think of that like as having infinite number of values. So histograms, scatter plots, line graphs, things like that. Sorry, we have one more practice problem. We have catalase, which is an enzyme. Anything that ends in ase is usually an enzyme. And pH. All right, so here you have, you want to see how pH affects the enzyme functionality. All right. And so here you have changes in pH and then the oxygen collected. So what type of graph do you think this is going to be if you're seeing how pH and the oxygen collected throughout time? All right, you should have said line graph again because time is continuous. And that should be it. So thank you for listening. Let me know if you have any questions because I know I did go quickly through this. I didn't want this video to be super long, but let me know if you have any questions.